request Krishnendu sir to begin the session. KG
Thank
sir.
you, Thank you, Maharaj. I think I'm audible to everyone.
Yes.
Yes, yes.
Okay, okay, thank you. So, very good evening to all of you. This is Krishnendu Ghosh, the faculty member of Department of Zoology, Ramakrishna Mission Vidyamandira, Belur Math. On behalf of this zoology department of this prestigious institution, it is my privilege to welcome you all in this international webinar on sickle cell hepatopathy and uncommon complication by Dr. Tirthadipa Pradhan-Sundd, Assistant Professor, Department of Medicine, University of Pittsburgh, USA. We all know that currently the entire world is going through a very inclement phase as the COVID-19 is raising its hydra heads, paralyzing all the academical, social, and ritualistic activities. However, we are all the positive believers. We are sure that the things will get better in near future. Our department and that of the college at large thus always tries to keep the flame of academic and research mentalities ablaze, driving the young mind of our students at par. Continuing this legacy, various webinars, online workshops have been catered by the institution at regular time interval, culminating a rich flavor of knowledge into enthusiastic naive minds. Thus, today is just another day of this academic festivity that I am pretty much sure is going to enthrall us, rising with the art of knowledge. To make it more reachable to a larger community, this is being streamed live in YouTube Live platform. So without much further delay, let us begin the journey with the holy chanting of Vedic hymn, remembering the divinity of the great holy trio, a spiritual enlightenment that will make a great inception. I'd request Sri Anik Mukherjee, student of third year zoology honors, who is going to do the honor. So over to Anik, please.
श्रीगुरुभ्यो नमः हरि ओम् सहनाववतु सहनो भुनक्तु सह वीर्यं कर्वावहै मा विद्विषावहै ओम शांति 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 ओम शन्नो मित्रः शं वरुणः शन्नो भवत्वर्यमा शन्नो इन्द्रो बृहस्पतिः शन्नो विष्णुरुक्रमः नमो ब्रह्मणे नमस्ते वायो त्वमेव प्रत्यक्षं ब्रह्मासि त्वामेव प्रत्यक्षं ब्रह्म वदिष्यामि ऋतं वदिष्यामि सत्यं वदिष्यामि तन्मामवतु तद्भक्तारमवतु अवतु माम् अवतु भक्तारम् ॐ शान्ति शान्ति शान्तिः मधुवातारितायते मधुक्षरन्ति सिन्धवः माधीर्नो सन्तोषधि मधुनक्तमुतोषसि मधुमत्पार्थिवगं रजः मधुद्गौरस्तनः पिता मधुमान्नो वनस्पतिर्मधुमार्गम् अस्तसूर्यः माधीर्गावो भवन्तु नः ॐ शान्ति शान्तिः ॐ पूर्णमदः पूर्णमिदं पूर्णात् पूर्णमुदच्यते पूर्णस्य पूर्णमादाय पूर्णमेवावशिष्यते ॐ शान्ति शान्ति शान्तिः हरि ओम तत्सत् श्रीरामकृष्णार्पणमस्तु।
Wonderful. So divine, so majestic indeed. Thank you, Anik. Now I'd like to request our Principal Maharaj, Swami Ekachittanandaji, to kindly deliver the welcome words.
Om Namo Bhagavate Ramakrishna. Respected Professor Tirthadipa Pradhan-Sundd, University of Pittsburgh, on behalf of Vidyamandira family and the Department of Zoology, let me extend my heartfelt namaskar, shraddha, respect, and good wishes to Professor Tirthadipa Pradhan. My respectful namaskar to our Vice Principal Maharaj, Swami Mahapragyanandaji, Professor Abhijit De, Head of the Department of Zoology, and other faculty members, the guest teachers who are attending this webinar, beloved students, and all the viewers of this session. Actually, during the pandemic situation, we have seen a tremendous upsurge, a bonanza in the arena of research in the biological field. As a layman only, I'm making this comment because of the current trend that all of us have witnessed. And given the fact that the Department of Zoology is a youthful, vibrant department, and it has got so much of vitality that also we have seen in the recent past. The boys have come out with astounding results in the competitive exams. So I'm sure that, as in the previous webinars, this one will also be contributing to a large extent to the enhancement of knowledge of this particular department. Once again, let me extend my heartfelt namaskar and respect to Professor Pradhan-Sundd on behalf of Vidyamandira family as well as the Department of Zoology.
Thank you, Principal Maharaj, for such a delightful note. Now I'd like to request Dr. Abhijit De, Head of the Department, Zoology, to kindly give the keynote address along with the official introduction of the esteemed speaker of this evening.
Thank you, KC sir. Good afternoon, everyone. This is Abhijit De, Faculty and Head, Department of Zoology, Ramakrishna Mission, Vidyamandira. Welcome all of you in this international webinar. Ramakrishna Mission Vidyamandira began its journey in 1941 following Swamiji's vision and comprehensive scheme of education. The college was upgraded to a 3-year degree college in 1960. Till now, this college has achieved many milestones. In 2013, the college was reaccredited by NAC with grade A. In 2014, the college was awarded the status of college with potential for excellence for the second time by the UGC. In 2020, our college ranked 7th in India and 1st in the West Bengal in NIRF ranking. Department of Zoology is the youngest department of Ramakrishna Mission Vidyamandira, established in 2015. From 2017, our department has started 3-year degree course in Zoology honors. Within this very short period of time, our first honors batch has come with colors. We are organizing webinar regularly from 2020, but today's webinar, entitled Sickle Cell Hepatopathy, an Uncommon Complication, is our first international webinar. Healthy red blood cells are round biconcave, which help them to move easily through the blood vessels. It contains hemoglobin, which carries oxygen to all parts of the body. Sickle cell disease is an autosomal recessive monogenic disorder that affects millions of people worldwide. Sickle cell anemia, which is a very common form of sickle cell disease, is caused by a homogeneous mutation in the beta globin gene, which makes the RBCs sickle-shaped. Sickle cell disease leads to hemoglobin polymerization, erythrocytic rigidity, dehydration, vasoocclusion, and premature hemolysis. The sickle cells die early, which causes a constant shortage in the red blood cells. Always, when they travel through the small blood vessels, they stuck and clog the blood flow. Vasoocclusion and the hemolysis are the predominant pathophysiological event in sickle cell disease that contribute to multi-organ damage. Acute and chronic liver complications have increased over the last few decades due to growing life expectancy of the patients with the sickle cell disease, but the molecular mechanism that promotes the sickle cell hepatopathy is largely unknown. Currently, there are no standard diagnostic criteria and standardized therapeutic approaches for the acute and chronic liver complications associated with the sickle cell disease. To understand the plausible molecular mechanism behind the sickle cell hepatopathy, we are really fortunate to have Dr. Tirthadipa Pradhan-Sundd as a speaker of this international webinar. Madam has completed her BSc and MSc in Zoology from University of Calcutta in 2003 and 2005 respectively, MPhil in Biomedical Science from University of Sheffield, UK in 2011. After that, she did her PhD in Biochemistry from SFU, British Columbia, Canada in 2015, and postdoctorate on the chronic liver injury from University of Pittsburgh, USA in 2018. Madam is currently appointed as Assistant Professor in the Department of Medicine, University of Pittsburgh. She has many publications in internationally reputed journals like Blood Advances, Hepatology, Cell Metabolism, and Gastroenterology, and many more. She has received many prestigious awards throughout her research career. With these few words, I like to conclude, and I request Dr. Tirthadipa Pradhan-Sundd to deliver her lecture. Over to you, Madam.
Ma'am, please switch on your microphone. We can't hear you.
Can you all hear me now?
Yeah. Perfectly.
Yes.
Thank you. So first of all, I really wanted to thank the organizing committee for giving me this opportunity to present at such an esteemed platform. I feel this is one of the greatest platform that I have presented my work so far, being from Calcutta, from West Bengal, and having that special connection we all do with Sharadama and Ramakrishna Dev. So I feel highly honored today, and I hope you all are safe and sound during this very unprecedented time, and hopefully we will all come out of it very soon together. And now, without much ado, I would like to just present my research work in front of the students who are the most important to me, as well as the esteemed faculties. So as it was introduced, my topic today is about sickle cell hepatopathy, which is an uncommon complication associated with sickle cell disease. So today, sickle cell disease is we all know it's an autosomal recessive monogenic disorder, and 70% of the patients are of African ethnicity. It's interesting that this happens due to a single amino acid change in the beta globin of hemoglobin, resulting in such drastic phenotypes. Shown here is a world map, and you can see the number of births in every continent associated with sickle cell disease. And I would like to point out that both in US as well as in India, sickle cell disease is frequent, and you can see that at least 25 to 50 babies are born in US who can have sickle cell disease, and in India the number is a little bit more. So this is really a drastic monogenic disorder. So there has been a few historic milestones associated with sickle cell disease, which I wanted to introduce introduced to the students. And the first milestone was in 1910 when Dr. Herrick first came across to a student's blood smear, and he found out that some of the RBCs were sickle-shaped or a crescent moon-shaped, and since then it was termed as a sickle cell disease. He got very interested in it, and he thought that this is something very novel, which we should proceed further, and I think he was so right. So the second important milestone was done by Dr. Pauling, where he first termed sickle cell anemia as a molecular disease, and we started understanding the genetic basis of a sickle cell anemia. And the third milestone, which is also, I think, is very important, was done by Dr. Ingram, where he actually showed that differences in normal human and sickle beta hemoglobins, and we kind of found out that this all happens due to a single amino acid change in the beta globin of hemoglobin. So today, as I mentioned, it's a global burden. According to a published report in Lancet, one of the very esteemed journal in the field of medicine, there are 3.2 million people who have sickle cell disease all over the world. And there are additionally 43 million people who are carriers of sickle cell trait. And that number is also very important because, as we all know, it's a genetic disease. So when two carriers marry, they can have the chances of having sickle cell disease patients or kids much more than a single carrier. There are already 176,000 deaths associated with sickle cell disease, and at present, although there are a few drugs, new drugs in the market, hydroxyurea, L-glutamine, and crizanlizumab are the FDA-approved drugs. The disease activity and mortality still remains high in sickle patients with or without taking these kind of medicines. So here I'm showing you what the situation of sickle cell disease worldwide. So these are nice pictographs. In 2010, you can see these were the estimated newborns with sickle cell disease worldwide, and the alarming factor is the bottom of the graph, where you can see in 2050 the numbers are expected to increase manyfold, and mainly this is due to two reasons. Number one is sickle cell patients are now, with a little bit of improved medicines and care, they are able to live longer, and number two, human evolution and migration to places of developed countries from underdeveloped countries. So we are expecting a sickle cell disease increase or explode in 2050, and this is a very high time to do research and understand the molecular mechanism and new therapeutic interventions associated with sickle cell anemia. Now, in our India, also sickle cell disease, as I mentioned, is kind of not uncommon. You can see in the map that places close to northern parts of India have sickle cell patients, and although sometimes they have a specific haplotype like Deep Punjab or sometimes the Arab Peninsula genotypes, which are less harmful in terms of the pathophysiology, still sickle cell disease is a burning problem even in India. And now coming to the complex pathophysiology that is associated with sickle cell disease. So you can see there it is very complex, and I feel this review here does a wonderful justice in explaining the very complex molecular pathophysiology associated with sickle cell disease. So I hope I'll be able to go with you and we will all understand what exactly is happening. So the first event is, it's a genetic disorder. One single amino acid change happens in the beta globin of our hemoglobin. So here glutamic acid is replaced by valine in the beta 6 position. That single amino acid change actually changes the hemoglobin activity, the hemoglobin behavior here. In the absence of oxygen, or when the oxygen saturation is low, these hemoglobins forms polymers and bundles. They also undergoes sickling, or they just become sickle-shaped. These sickle-shaped hemoglobins now, or the sickle-shaped RBC, I'm sorry, the sickle RBCs are now unable to move as freely through the hepatic or through the any vessels of our body, and they kind of accumulates with other neutrophils, endothelium, and platelet, whatever you have in the blood vessel, the other types of cells, and these accumulations actually impairs the blood flow, as one of the speaker very nicely explained. So this is termed as vaso-occlusion, where there is just accumulation of RBCs and neutrophils, endothelium, and platelets. Vaso-occlusion then leads to ischemia reperfusion injury. Because of impaired blood flow, you don't have enough oxygen in that tissue. You have ischemia reperfusion organ injury, resulting in sterile inflammation. Now, if you look at the other half of the circle, the same sickling of RBCs, now the RBCs are unable to perform their function. Their shape has changed. Their many of the molecular properties have changed. They undergo hemolysis or burst out, and this hemolysis then causes endothelial dysfunction, resulting in accumulation of cell-free heme, which acts as a damage-associated molecular pattern and further kind of promotes the sterile inflammation. So basically, if you look at it, the predominant events are vaso-occlusion, sterile inflammation, hemolysis, and endothelial dysfunction, which all together causes a prolonged sterile inflammation constantly, which is going on in different organs of a sickle cell disease patient. So, when we talk about the clinical complications of sickle cell disease, you can see they are manifold. Almost all the organs in our body can be affected by this disease. And some of them are more common and very highly studied, such as stroke or acute chest syndrome or cardiomegaly and pulmonary hypertension. And some of the other phenotypes are right now coming more into limelight. And one such particular problem that is the focus of my research is the liver. So before going to the liver, as a picture speaks 1,000 words, I wanted to show some real pictures of how exactly all these different organs are affected in sickle cell anemia. And you can see they have very commonly associated with proliferative retinopathy, where your retina stops functioning. Pain is always associated with sickle cell patients. They starting from as early as like 6 months or more, a kid can actually experience pain, and this pain related to vaso-occlusive crisis can actually cause organ damage, such as joint replacement or chronic pain of AVN. They have a very large spleen, which is almost in the patients, the spleen is non-functional. So the splenic infarction and sequestration again causes pain and acute anemia. They also have leg ulcers, which are pretty painful. So basically, if you have sickle cell disease, you live with pain, and medicines are mostly directed to somehow reduce the pain that these patients go throughout their life. Now, coming back to the liver. So, as my title says, it's relatively uncommon, but if you see the numbers, there are millions of patients with sickle cell anemia, and 10 to 40% of the hospitalized patients can have liver involvement. And here, previous reports have shown that the liver can have acute injury as well as chronic liver injury, and it's a lot laundry list of what other liver complications you can have associated with sickle cell disease. You can have iron overload related chronic liver failure. You can have hepatic crisis. You can have cholestasis. You can also have hepatic sequestration, gallstone, which is kind of related to the hepatobiliary pathophysiology. So it's a long list of things, and whether all of these are related to the sickling process or there are independent factors causing liver diseases in sickle cell anemia patients are not very well understood. So one last slide before I actually go to my research and show you some actual data is it's like so it's good and bad. The good thing is right now, because of these wonderful like therapeutic interventions possible today for sickle cell anemia patients such as hydroxyurea, crizanlizumab, L-glutamine, their life expectancy has increased over the last decade, but that also has caused reports or incidences of more organ injuries, because I feel the body goes through repeated hemolysis, repeated vaso-occlusion for a prolonged period of time, and now instead of just having maybe acute chest syndrome or pulmonary hypertension, now the liver and kidney and other organs are coming more and more into the limelight. So in our lab, we actually do basic translational research, and in collaboration with wonderful faculties here at VMI, we try to understand this disease starting from looking at a humanized mouse model. So why I'm calling it a humanized sickle mice? Because here, the sickle human beta globin is actually replaced with the mouse beta globin. So the mouse doesn't have any mouse beta globin. It has the sickle human beta globin, and numerous reports have wonderfully shown that this humanized Townes mice can very well recapitulate the blood parameters which are historically associated with sickle cell disease, such as reduced hemoglobin and increase in neutrophils and increase in reticulocytes, as well as reduced hematocrit. So this is a very wonderful murine model to study the molecular mechanism that is happening in sickle cell disease. So the first question we asked, because in our lab we have this wonderful tool to kind of view in real time what is happening in the blood, what is happening in different organs in terms of tissue interactions. And what we do is we just do intravital imaging. So here is a schematic of the intravital imaging, and you can basically term it as an ICU for a mouse. So here we try to keep the blood pressure, the temperature, and the flow of blood as constant, as normal as possible, basically not giving the mouse any kind of stress as much as possible. And then you inject dyes through intravascular or IV injections, and basically you image in real time what is happening inside your body. So these are some of the intravital videos. So first of all, what I wanted to check is how is the blood flow, hepatic blood flow in the sickle mouse, and how is it different from a control mouse liver. So here I'm showing 3 examples of a control mouse liver, and you can see I have injected Texas red dextran in the hepatic through IV, and they actually show you how is the blood flowing through the hepatic sinusoid. What to kind of note here is we do not see any gap in the sinusoidal field. Blood is flowing continuously. We do not see much or any abnormality in a control mouse. Now, if I go to the sickle liver, I hope you can appreciate here that these sickle mice, and this is all at baseline in the liver without any kind of treatment, what you can see is there are huge regions which kind of appear as dark spots, but there is a stasis of the blood flow, or actually you can see, if you use the right term, you can say vaso-occlusion is seen at baseline in a sickle mouse liver. So we were kind of very excited because that actually says that the mouse model can wonderfully recapitulate the predominant pathophysiologies that are seen in patients. So we wanted to now characterize why at baseline there is impaired blood flow and how to understand the molecular mechanism behind it. So for that, having a training in pathology, we went back to old school pathological techniques which are extremely relevant even today. So what we saw is, as compared to control mouse liver, the sickle liver was darker, and it also had white spots suggestive of ischemia, reperfusion injury, or you have like oxygen depletion in particular regions of the liver. And the liver was also a little bit larger, as shown by the liver weight by body weight. So large liver, normally if the liver weight by body weight ratio is a little aberrant than the control, it's kind of indication of impaired function of the liver, which is associated with many liver diseases. Then we looked at the blood serum parameters, and what we could see is many of the markers such as triglyceride or glucose were normal or comparable between sickle and control mouse, suggestive of, like, it's not that a completely non-functional liver, they have some of the function intact at least. And when we looked at injury markers of the liver, ALP, ALT, and AST, which are very well-known injury markers associated with liver diseases, we could see that in sickle mice, again, at baseline, there was an increase in ALT and AST and slight increase of alkaline phosphatase, or ALP. Interestingly, when we looked at bilirubin, we all know that bilirubin increase is associated with liver diseases. Both total and direct bilirubins were high. It was moderately high but significant in the sickle mouse liver. And finally, we just ran through a plethora of like pathological like antibody staining, and basically the take-home message is what we found, that the sickle liver is injured. It has a liver injury, increased fibrosis by serious red staining, and alpha-SMA shows you the activation of myofibroblast. Tunnel staining just showed that there is a mild increase in hepatocytes which are undergoing apoptosis or cell death. And we found a strong increase in a macrophage marker, F480, which is a hepatic Kupffer cell marker suggestive of sterile inflammation. So basically what is known in patients that, in like studies using other tissues and organs, that they will have sterile inflammation and they will have injury, which we could kind of recapitulate in the mouse model at baseline. So normally the humans had to go through a crisis, like it can be a pain crisis, it can be high altitude, it can be dehydration, but it was very interesting to find in these mice, we did not challenge them with any kind of hits. They just had this at baseline. So then we again came back to our intravital imaging, and now you can already understand that this is one of our favorite techniques. So here, compared to the previous videos that I have shown you, I am actually showing you two different dyes. One is the Texas red dextran, which is obviously marking the blood flow through the hepatic sinusoids, and the green one is a specific dye called carboxyfluorescein, or CF, that actually has a unique properties. When you inject it from the blood, it first goes to the hepatocyte, and from the hepatocyte very quickly, very transiently, it eventually goes to the bile duct or the canaliculi, where your bile is stored. So these are two control liver intravital imaging, and you can see that initially there will be some CF staining marking the hexagonal hepatocytes, the borders of the hepatocytes, but as we go forward, all the CF kind of completely accumulates to the duct or the bile canaliculi where it's destined to be. So we thought, can we track the movement of CF in sickle mice, and can we see if there is any difference? Because sickle cell anemia patients, a group of, like, again, a particular population were also associated with cholestasis, which is a ductular injury and malformation of bile duct, which kind of results in impaired bile flow and bile trafficking. That can cause an increase in bilirubin level as well, along with the main reason of hemolysis that is happening in sickle cell anemia. So here we actually found a fantastic phenotype, and I hope you can appreciate. So what is happening at any given time point when we examined was that the CF was kind of not completely leaving the hepatocytes. So there are some shadows which persistently present in the hepatocyte, suggestive of either two things. Number one, that the bile transfer is delayed, or there could be some regurgitation of bile from the duct to the hepatocyte. So we were very excited. This was extremely like novel phenotype, and it was very consistent that there is a delayed bile transport in sickle mice. So next, we wanted to molecularly characterize. These are just the images, not the videos, and I hope you can appreciate from just comparing the two panels that in control, CF actually is very prominently present in the ductular area. We don't see much of a shadow of CF, which we see in the sickle cell anemia mouse. So before further characterizing this phenotype, we wanted to check whether these are actually consistent with what we see in patients, because again, this is a real disease. So you can see these are all patient serum samples which we got in collaboration at UPMC, which is a fantastic place to do sickle research. And you can see that even in patients at baseline, they could show some increase in liver injury markers, total and direct bilirubin, although it was not very highly significant over the roof, but it was present. And they also had a little bit of increase in the serum bile acid, which can actually indicate the phenotype that we saw in mice, which is a delayed bile transfer. So we thought, okay, this is an interesting phenotype. Why don't we characterize it more and find out what exactly is happening? So first phenotype that was like kind of the most prominent in our hand was that we saw significant sterile inflammation. So here I'm showing you IHC or immunohistochemistry images of markers such as F480 and CD45. They are actually markers of the hepatic macrophages, and you can see compared to control in the left, in sickle mice, these markers were significantly enriched. And suggestive of sterile inflammation. When we looked at and the regions close to the ductular area also looked kind of as if they are having some distortion. So we also looked at markers of markers which are known to regulate cellular senescence. And we were very happy to find that P21, which is a marker of cellular senescence, was increased or enriched in sickle mouse liver. We kind of confirmed it with IHC as well as IF, as you can see here, and co-localized with CK19, which is a marker of the ductular cells of the cholangiocytes. Further, we just confirmed that this is really a phenotype because the mice can be very variable in nature, and that by qPCR as well as Western blot, that they had an increase in cellular senescence as well as an increase in sterile inflammation shown by increase in cytokines as well as macrophage markers. So all these kind of hinted us to look at NFkB pathway. And for the students, NFkB is actually a very well-studied pathway which regulates inflammation. And regulators of NFkB, such as TNF-alpha or IL-1 beta, were found to be increased in our sickle mouse liver. So we thought maybe there is an increase in NFkB pathway. And to prove that, we first did an RNA-seq of the whole liver. liver. And shown here in the heat map is the result of the RNA-seq. And you can see we just cherry-picked the genes associated with NF-κB signaling, and many of them were actually highly activated in a sickle liver at baseline. And this was further confirmed by looking at the qPCR, by looking at the NF-κB P65 staining, which is a marker or readout of NF-κB activity in the liver, and a Western blot of phospho-P65. We also looked at a few markers by Western, and all of them were kind of activated in sickle liver. So we thought, okay, NF-κB activation is kind of, you can anticipate this. What else can we find out from this result? And very interestingly, along with its many roles in the liver or in any other tissues, NF-κB in the liver and intestine has been shown to regulate FXR, which is a master regulator of bile synthesis, trafficking, and transport. And remarkably, when we looked at FXR pathway, what we could see is that FXR is down-regulated in the sickle mouse liver. I'll not go into very intricate detail of all the panels. So basically, after this inactivation of FXR, what happens is the bile acids are accumulating. There is delayed bile transport. And very interestingly, in the lower panels, especially if you look at the pie charts, here we did a bile acid analysis. So in our body, there can be many different types of bile acids. Some of them are more toxic than others. And what we saw is in sickle liver, the bile acid composition was different than a control liver, making their bile more toxic. So this was also very interesting. And finally, the final approach for every lab is to somehow try to ameliorate some of the phenotypes. So we did an NAC treatment, which is known to inhibit NF-κB activation. So what we did here is we took a 4-week-old mice. We did 12 weeks of NAC treatment in drinking water, and then we harvested the mice. We show that NF-κB is a little bit down-regulated here by looking at NF-κB P65 as well as markers of the NF-κB signaling pathway. And now when we kind of analyze the liver injury, we were, this was like a not all or none. So we saw a slight reduction in liver injury markers and a significant reduction in bilirubins, as well as the liver tissue as a whole looked a little better. And these are the images of the intravital imaging, and you can see that in sickle mice, shown by this red dotted arrow, there were these regions of vaso-occlusion or stasis of blood flow that was also to some extent ameliorated after NSC treatment. And we further proved that NSC can also regulate FXR. So NF-κB FXR, this interaction was further confirmed using the NSC treatment. But we have to be very careful in saying this repeatedly, that we did not see a complete or a drastic amelioration of this phenotype. So from the first part of my study, this is our working model, and this is kind of characterization of the liver phenotype. So what exactly happening here is, remember those stainings where inflammation was significantly up. So there is a baseline inflammation in sickle mouse, which leads to activation of cytokines such as IL-1 beta, TNF-alpha, which activates NF-κB. Now here, NF-κB goes to the nucleus and it inhibits FXR. As a result of that, the transport of bile from hepatocyte to the canaliculi is affected, because many of the transporters such as BCAP, ABCG5 and 8 are regulated by the FXR signaling. So that can explain why we see a delayed bile transport, because your gates are all closed, or many of them are closed. So somehow there is a traffic there. The delay can actually signify why there is cholestasis in some sickle patients. So, a summary of the first part, I hope you all agree with me that this humanized mouse model shows hepatic ischemia reperfusion injury at baseline by intravital imaging. We saw that these mice had liver fibrosis, ductular reaction, and hyperbilirubinemia. And most importantly, they had a delay in their bile flow. When we tried to molecularly characterize this delayed bile flow, we saw activation of NFkB signaling, which impaired FXR pathway, and blocking NFkB using NAC can provide some amelioration of the liver injury. So this was all good. Now, I would also like to briefly touch on one of our very recent publication, which was also like very unexpected. So this was published this month in Blood, where we actually showed that P-selectin deficiency, now I'll come to P-selectin in my next slide, it promotes liver senescence in sickle cell disease mice. So why did we mechanistically to address this question, sorry, I think there is
I think
bear with me for a second.
yeah.
So I'll just go to the next slide. So what exactly happening here is P-selectin inhibition by crizanlizumab is a known therapy which is FDA approved now, which improves the pain crisis in sickle patients wonderfully. And these are published in very high impact journals, and P-selectin is used as a regulator to regulate vaso-occlusion and vaso-occlusion mediated pain crisis in sickle patients. In collaboration with Sundd Lab, we had this P-selectin deficient sickle mouse, and the question that we asked was what exactly happens in this mice liver. So remarkably, when we looked at vaso-occlusion as seen in other organs like lung, we could see that the regions of stasis, or the dark regions where the blood flow was impaired, actually got ameliorated when we depleted P-selectin. So this was a good news that your ischemia reperfusion injury can be somehow ameliorated with P-selectin. However, when we looked at the overall liver pathology, what we see is that P-selectin is unable to improve the chronic liver injury that is present in the sickle mouse. So here, by Prussian blue staining, I'm actually showing you the iron accumulation in the whole liver, and I hope you can appreciate that in P-selectin knockout sickle mouse, compared to sickle alone, there were more iron positive blue vesicles, which we quantified, and it came significant. When we looked at bilirubins, they did not show a reduction in after P-selectin treatment. And when we tried to mechanistically look into this by looking at the leukocytes, what we saw is remarkably after P-selectin depletion, the leukocytes were significantly reduced in the liver, which is shown by qPCR as well as by IF staining of Ly6G, which kind of increased the hepatic fibrosis and markers associated with hepatic fibrosis such as TGF-beta, collagen, and TIMF1. So basically, this is very interesting, which kind of suggests that long-term therapy of crizanlizumab can be deleterious or can be having some side effects in the liver, increasing their iron accumulation and causing defects in iron trafficking mechanism. So this was our working model for the P-selectin story, which I kind of very briefly touched upon. So here we are showing the role of sterile inflammation. So remember, we started with sterile inflammation, which plays a very important role, but here we show that the leukocytes from the blood, when they move into the liver in a sickle mouse, they actually play two different roles. It's always like nobody is 100% white or nobody is 100% black. So one of their role is to promote ischemia, which causes chronic liver injury, and the other role is good, which is resolution of inflammation. So when you do not have these leukocytes due to p-selectin deficiency, what you have is ischemia is somehow like improved. However, the resolution of inflammation was completely impaired, which increases the overall chronic liver injury, increases liver senescence, and hemolysis-driven liver injury in both these models were unaffected. So this was very interesting and in a way unexpected. So this brief report was also kind of accepted as a cover article of the Blood Journal. So we were very excited. So finally, this is my take-home message slide, and I hope we all were able to kind of understand that sickle mice and sickle patients, they both have some liver involvements. Especially the humanized sickle mice, using intravital imaging, we were able to show that they have this ischemia reperfusion injury, where there is a huge region of blood stasis in the liver. They also showed liver fibrosis, tactilar reaction, and hyperbilirubinemia. Using in vivo live imaging, we were able to show that the bile flow was delayed in sickle mice, which is due to activated NFkB signaling, which impairs the FXR pathway. So one of our aim going forward is maybe can we inhibit the NFkB signaling as well as somehow activate the FXR pathway. So finally, p-selectin deficiency we showed that protects from hepatic vaso-occlusion. However, it does not prevent the chronic liver injury. Furthermore, finally, we also found that there was increased cellular senescence in the liver of this sickle cell disease mice. So overall, this is one of the main focus of our study, just understanding different kinds of molecular mechanism that we can imply to somehow ameliorate the liver diseases which are associated with sickle cell anemia. And some of the future directions which we are currently doing and want to do more in future is tease out the role of hemolysis, tease out the role of vaso-occlusion and maybe endothelial injury in the sickle mouse, and how each of them they together can contribute to the liver injury. We are very interested in finding out the iron metabolism in the liver in sickle cell mouse, because they are constantly having hemolysis, which is releasing heme, resulting in accumulation of iron, and what exactly can be done to somehow impair this process and block the chronic liver injury. We are also interested in understanding the potential role of hepatic macrophages as well as the crosstalk between hepatocytes and macrophages in sickle cell disease. And as p-selectin, we also want to test, because liver is one of the most important organ for its extreme resilience against any kind of stress due to its regenerative potential, but liver damage can actually affect other organs at a very fast manner. So how many of these FDA approved drugs which are in the market can actually affect the liver injury in sickle cell anemia. So with all that, I'd like to thank many people, and this is here in VMI, my mentors, collaborators, my lab people, and as well as the department I belong to, and I thank the funding sources as well. And finally, I really wanted to thank all of you for giving me this opportunity. I wanted to just pay my homage to Ma Sharda, and I remember that my grandmother used to say that she is a living embodiment of the goddess of knowledge, so Ma Saraswati. So I hope that she accepts my homage, and I sincerely thank my parents. They are listening from somewhere, and thanks to you all. Thank you.
Thank you, madam, for this wonderful lecture. And now I would like to request Dr. Debarun Acharya, faculty member, Department of Zoology, to cater the Q&A session. So over to DA, sir.
Thank you, KG sir. And I thank the speaker for her wonderful talk. So now I would like to read out the questions from the audience. So the first question is from SDG sir. So the question is there any relation with sickle cell patient and malaria? I heard that sickle cell patients never suffers from malaria. Is it true?
Yes, that is a wonderful question. And this actually brings a lot of history of this disease. So sickle cell disease actually, there are reports that it evolved as a defense mechanism against malaria. So obviously in Africa and many other developing countries, including India, there is a prevalence of malaria, and sickle cell anemia patients are somehow protected from malaria. So this was a defense mechanism to protect themselves from this deadly disease. So yes, they are very interconnected. There are many publications which says that this actually evolved to kind of fight against malaria. But in today's world, in US, there is no evolutionary advantages of having sickle cell disease, because we do not have malaria now. So now it's basically something which once actually was helpful to you to survive is not playing the same role anymore. Yeah.
Okay. So the next question is from Saurabh Bhattacharya, which is, what did you use as positive control while doing bile duct analysis? And the next one is, if Mr. X wants to design a drug, what should be his preferred target, the effexor pathway or something else?
Both are very good questions, and I'm very excited because the new generation is very intelligent. I feel they're always ahead of me. I have a 4-year-old, and I can see the evolution is happening so fast. So for bile duct experiments, controls were always AS mice. So these mice have one copy of the sickle beta globin, and they are the carriers. And in our hand, AS mice did not have any kind of defect. So it's actually a litter-met control, the best control that you can have. And for the second question, I don't think I'm the right person because I'm not from the field of drug discovery. But related to my research, I would say we have done some treatments associated with NFkB and associated with FXR. So I would go more towards NFkB because, again, this is all our personal experience. FXR was very mild, and we did not see a very strong phenotype. Yeah.
So the next question is from PD sir. So he wants an enlightenment about the current therapeutic development and gene therapy potential for curing this disease.
Yes. Here I heard my esteemed colleagues present that we are going through a sickle cell disease gene therapy tsunami. So there are a lot of things that are coming out now, and gene therapy is again it's like a very long-term approach, but there have been wonderful advancement in this field. You can actually read, or I can send a few papers. So this is going on very promisingly. There are therapies related to hemopexin that is going on in some companies, and there are crizanlizumab was also very successful, L-glutamine, and so these are not gene therapy, these are medicines. In gene therapy, it's slowly growing, but I guess we will see something soon, if not too far away. Yeah.
Okay. The next one is from Shubham Shaha, who wants to know that what are the limitations of the present drugs and their target sites, which we like to eliminate in future studies or future works.
Yeah, these are all wonderful questions. So What we have seen here from the like the doctors that have actually dealt in real time with these patients is this is kind of some obviously hydroxyurea, which is the one of the best drug for sickle cell disease anemia patients, have been very successful. Even crizanlizumab is also successful. So some of them can have some side effects, like as I showed in my p-selectin part, that although it's improving the hepatic ischemia reperfusion injury, the chronic liver injury can be worsened. So maybe combination of drugs, maybe p-selectin or crizanlizumab along with hydroxyurea, and all these are going on right now. So a combinatorial approach might be more useful. Again, I'm not a physician, so I hope I was able to answer it more. So a physician will be the best to correctly answer the drug part of this project. Yeah.
Yes, thank you. The next one is from KG sir. So he wants to know whether any particular type of macrophage polarization involved in this hepatopathy. And the next one is any epigenetic modifications related to p-selectin pathway, whether it is there.
Yeah, both of them are fantastic questions. The first question answer is yes. When we looked at macrophage polarization, we just looked at specific markers of M1 and M2. And what we see is very interesting. At baseline, there is like both M1 and M2 are like equally activated. But when you treat them with some kind of stress, such as LPS, obviously you'll see, and this is published, that there is more M1 activation. This was very interesting, and this is an unpublished data which we are trying to publish, that at baseline, without any treatment, there is a like a combinatorial approach, like both the population M1 and M2 are activated in sickle mice liver. The second question is again very cool. I don't know how to answer this. So obviously there can be epigenetic changes when you knock down p-selectin in the sickle mouse. But remember, in the patients, they do not have this long-term genetic like deletion. They just have p-selectin inhibition as a drug, crizanlizumab. So the epigenetic changes might not be extremely relevant in human patients. However, if we find out anything interesting using our murine model, that can also be very useful.
Okay, thank you. The next question is, do these sickle cells stimulate the tumor-associated cells in the liver and direct to hepatocellular carcinoma?
Good question.
Yes, can you just briefly elucidate the mechanism?
Yeah, this is also a good question. So there are not a lot of reports of sickle patients undergoing hepatocellular carcinoma. So I guess, and when we looked at the livers in the using IHC of the mouse liver tissue, we did not see any tumor formation, but that does not rule out that they will not have. I think what can happen is if they are constantly facing some kind of a challenge, which they can, like if a person is constantly like having ischemia reperfusion injury, or maybe in extreme increase in iron accumulation due to maybe some particular mutation, then I think that sickle patient can have like hepatocellular carcinoma. Right now, it's not very frequent according to the literature which is published.
Okay. The next question is there any relation between hepatocellular carcinoma and sickle cell? And okay, it is already answered, I think. And can liver cancer have an effect on this disease progression? So this is
very good question. So yeah, what one can do is, this is very interesting, that there are drugs or there are chemicals like CCl4 per se, which are known to cause hepatocellular carcinoma after a point, or DEN treatment. So we can compare in control and sickle mouse model and see the initiation and progression and maintenance in these two groups. And maybe it is interesting. Yeah, this is a good question. There are no studies which have already done it. Like, yeah.
Okay, thank you. The last question is, what are the causes of p-selectin deficiency? Can you elaborate a little.
Yeah, so P-selectin deficiency, I think I was not able to explain it properly. I should have added a slide. So P-selectin is actually a marker of the endothelial cell and platelets. So it's a cell adhesion molecule. It helps in adhering or adherence of cells with, like, suppose if you have neutrophils, monocytes, then P-selectin, it has a ligand, PSGL1. The ligand will bind with P-selectin, which will kind of promote the cellular adherence. So deficiency of P-selectin, normally what happens is, like in sickle cell disease, you'll have increase in P-selectin because remember, they have the sickled RBCs which are unable to move properly from the vessels, causing accumulation with other neutrophils, monocytes, and other endothelial cells. So absence of P-selectin can be maybe associated with a particular disease, but in this case, this is the opposite. Its increase in P-selectin is seen in patients and other mouse models. That's why there was a approach to deplete P-selectin and then see the effects in patients and mice.
Okay, so thank you. There is no more questions. So I would like to request KG sir to take over. Thank you for this wonderful talk.
Thank you.
Thank you, dear sir. And really, what a session we just went through. And it is indeed splendid. And it is really very hard to end up sessions such as this. But however, we should end so that a new beginning should be there, right? So now I would request our Vice Principal Maharaj, Swami Mahapragyananda ji, to give.
Am I audible? You can listen to me, right?
Yes, Maharaj.
So just echoing to the words of our dear KG Sir Krishnendu, I must say that this session was so interesting. It was just like, it seems to me, it's like a flow. Very interesting. From the beginning to the end, I was there. And I can't resist myself to quote, obviously little philosophical, from Tagore here. In one of his songs, he wrote, Madhuro tomar shesh tena pai. So it is like that. And I don't know how many of you know that Dr. Pradhan is very, very fluent in Bengali. She was born and brought up here in Calcutta. So, well, the thing is, if I say, it will be inadequate to say that this is something marvelous, something wonderful, whatever. But the thing is, if I look back, when we started the journey, especially in this Vidyamandira, the abode of learning, an outcome of the epoch-making thinker Swami Vivekananda, who himself coined this term Vidyamandira, and once he dreamed that it will become an epicenter of knowledge, where the people across the globe will come and stay together and share their ideas. So when I look back, in nearly 7 years something I'm here. When we started our journey, we initiated the Department of Zoology. The idea was to do some capacity building. From the very beginning, the entire team of us were very keen that the students should have the real thirst for knowledge, and they can enrich themselves. And I'm sure a session like today will give definitely a wonderful impetus to that idea and that thought. And on behalf of Vidyamandira family, I extend my heartfelt thanks and gratitude to Dr. Pradhan to be with us today and share her ideas. And I fervently hope to have her, and definitely I already told her few of her colleagues in this way, in such kind of webinar and association. So I really don't have my language to extend her thanks. So, and today, all our faculty members of Zoology Department, including the HOD sir, Dr. Abhijit Dev, and Krishnendu sir, Debarun sir. So from the very beginning, from the inception of this webinar, they were with us. So I thank them also for their participation. Of course, they are the part of the host, but it is customary to convey thanks. And especially our Onik, who began the session invoking the Vedic chanting. My best wishes to him also. And last but not the least, this entire platform, the streaming is going on with YouTube. So just behind the screen, we had our Ramakrishna Mondal is one of the faculty members of Department of Bengali in Ramakrishna Mission Vidyamandira, and Jit Maharaj, Brahmachari Ishatma Chaitanya. So I thank the participants who are with us today in YouTube platform, and especially I would like to extend my heartfelt gratitude, pranams to our teachers, acharyas. We had with us today Dr. Shubhit Dasgupta, and just like our acharya, he was one of the mentors. He was our acharya, he is today, and sir is one of the mentors of this department. We have today with us, we thank to be here, Dr. Pratap Kumar Das is an eminent scientist and a very senior faculty. Right now, Dr. Das is also with us, so I thank him personally, and Dr. Ashit Shorkar, the senior most faculty of the Department of Microbiology, and all other teachers of Microbiology department, and the students, especially Zoology and Microbiology. So thank you everyone, and hopefully, really, it's an amazing one. For last nearly 1 hour, we almost forgot where we started and where we end. So once again, thank you everyone, and thank you, Dr. Pradhan.
Thank you so much, Mahapragyanandaji. This is all your effort, and I'm really happy that you are going towards your mission and enlightening many people, and you'll do more as you go through. Thank you so much from the bottom of my heart.
Thank you everyone. So I think it is now official to call this thing off. So
Yes.
Thank you so much. Thank you.